Excellent. I'm going to introduce you here.
Okay, I'll get out of your way.
All right, let's get started. Thank you all for coming. This will be our last Quantitative Systems Biology Center seminar of the school year. And first, before I introduce our speaker, I want to give a huge shout out to Becca Sykes, who has been coordinating the QSBC seminar series all year. She's done an amazing job helping coordinate all the speakers, and she does everything. So I am greatly indebted to her, all of her hard work. So thank you. Yes. So without further ado, though, I'd like to introduce today's speaker, Paul Macklin. He is now an associate professor and the associate chair of the Department of Intelligent Systems Engineering at Indiana University in Bloomington. And he was recently appointed to be the new Associate Dean of Undergraduate Education. So fantastic advancement. He got his bachelor's degree in a dual degree, actually, in mathematics and German from University of Nebraska, and went on to get a master's degree in industrial and applied mathematics at University of Minnesota in the Twin Cities underneath the tutelage of John Lowengrub. Then he followed John to UC Irvine, where Paul got his PhD in computational and applied math, and then has done several different positions after that. And he took a position as an assistant professor of health informatics at the University of Texas Health Science Center under Vittorio Cristini, and then moved to University of Dundee for a year and became a lecturer in the Division of Mathematics there before he became in his full first tenure track position at the University of Southern California in the research medicine in the Center for Applied Molecular Medicine at USC as an assistant professor, and then took his position at University of Indiana in 2017, I believe, and rose his way through tenure and all the way up to his current position. He's done some amazing work with this agent-based modeling approach, PhysiCell, as the instrumental designer of this entire package that is just a phenomenal resource. And I'm sure he'll showcase all of its functionality today in his talk, using agent-based modeling as a virtual laboratory. Thanks, Paul.
Well, thank you for the invitation, also for the biography. I've actually forgotten some pieces of it, so thank you. It's a great pleasure to be with you and be with old friends and new friends today. Vito, it's been a while, and it's just fantastic to be back here in Nashville. I did. It wasn't too bad. So it's really great to be here and to meet with new people. Just had a wonderful conversation in a few this morning. And so, yeah, my path is a bit of a weird, windy one, where I started off, German has not quite been the most useful thing, but it's been great for travel and for collaborations. And, kind of from that math perspective, my PhD work was actually primarily in continuum mathematics, fluid mechanics, moving boundary problems, a lot of numerical solution techniques. And then I took my first job at the University of Texas Health Science Center, thank you for reminding me, and took a look under a double microscope at a breast pathologist and said, we need a different approach. We can't match any of this stuff to these, 5-parameter models that we've been doing. So that's took me down a spin towards agent-based modeling to start really getting down and describing cells as the fundamental unit of our model, as the agents that move around and do things and respond to their environments and kick their environment around. So I'll start introducing that perspective here. But being that windy path, I have my continuum roots and my discrete roots, and then having been embedded in the foreign language of medical schools, come to appreciate and love the biology. Originally, as an applied mathematician, I thought, well, this is interesting. I'll stay in biology for a few years, and then move on to something else. And of course, biology always surprises you every day, and you don't run out of interesting things to do. So here I am, like 20 years later, still doing biology. So we're really grateful to have had some funding from a variety of sources, perhaps forces, including the NIH is certainly a force. DITRA is definitely a force. They talk about the warfighters all the time. Of course, philanthropy, and we're really grateful for that. So something that really gets me interested as a mathematician looking at these biological systems, looking under microscopes, looking at pathology slides, is really starting off with, that, biology is complicated, but in some sense, you can abstract it back and say, out of all the zillion things that you see, that is all emerging from a fairly finite list of things. That cells have a finite list of fundamental behaviors. They can divide, they can change and grow volume, they can differentiate and change type or mutate. Of course, they can die, they can consume things in the environment or sample things in the environment and metabolize them and then secrete things back out into the environment, which can act as chemical signals. Of course, there's all their internal signaling cascades, but fundamentally, they're growing and dividing, dying, secreting signals and sensing them and moving around. And then you add in, a handful of pairwise interactions. They can stick to each other, they can push each other around, they can eat each other, and find other ways to talk to each other by contact. And then you put them into an environment with physical constraints. They have chemical gradients and mechanical barriers. And somehow from this finite set of interactions, put together a population, 10 to the 5, 10 to the 6, 10 to the some power, and you get the emergent biology of all the messiness that we study, of the complex systems of cells chasing each other around and becoming immune to therapies and building structures and somehow maintaining them and not falling apart. And it's like the more you learn about biology, the more amazing it is that we don't just spontaneously fall apart, and somehow they're overcoming entropy and all these things to build and maintain these complex systems. And then in disease, of course, some of these edges start malfunctioning and we get the emergence of diseases and disorders. And so multicellular systems biology says, how do we go from these interactions, these individual behaviors and small set of interactions behaviors in these constrained environments, and get to the complex systems that are you and me, and understand how these behaviors tie together. And of course, we would like to go not just systems biology and understand the systems, but we want to go to systems engineering and engineer the behavior of these systems. Say, if I can change this interaction, that interaction, this behavior, can I steer the emergent community behavior to get to a state that we prefer to be in? One that's preferably not autoimmune, one that's preferably not hyperproliferative or degenerative or something else. And so this being a complex system, we need model systems to understand it and attack it. And so biologists have, of course, a handful of model systems. Of course, as a mathematician, I was amazed to learn that all models were not mathematical. I thought that they were synonymous until I got out of my little bubble. And that model can mean a broad variety of things, ranging from cells in a dish to 3D printed systems to engineered systems, all the way to computational numerical models like we do. So for us, model systems that we're going to use as our virtual laboratory will be these agent-based models that we'll introduce in just a moment. But like any model system, the idea is to get to something we can get our hands around and perform experiments in to start teasing out the behaviors of these systems and saying, if we think our hypotheses are these, what are the emergent behaviors? Does it match what we believe? And if not, what are we missing? And start posing new hypotheses until we can get a better match, and then maybe plan a better experiment to probe those new hypotheses. So let me start kind of narrowing back to a conceptual model before we go to the computational models. That we're trying to think more and more of these multicellular systems as communication networks. To say that you have a sender and a recipient. And the signal can be anything. It can be basically a stimulus that can elicit a response. So a sender might secrete a chemical diffusing signal. These are the ones we're more used to. The signal could also be mechanical pressure. It could be contact with one another, either through receptor ligand pairing or just by the notion of detecting that contact in mechanics. But that signal then from the sender cell it becomes a stimulus to elicit a response in the recipient. And the responses can be anything. It could be changes in cycling, changes in depth, changes in migration, motility, even changes in volumetric growth rates that might start or stop growing, changes in metabolism, changes in consumption metabolic factors and other factors, changes in secretion of signals. So there can be a feedback signal and behavior, changes in predation, mutation, cell type changes. So it's this bizarre combinatoric space of possibilities, and we start adding these networks together. And so we're trying to think of this in terms of who is sending a signal, who is receiving that signal, and what do they do in response to the signal. And if you can put these things together, you're starting to get at the ideas that we want to do in agent-based modeling. And so it turns out that they are really well suited to this kind of this framing. And so agent-based models is a software concept. So if anyone's done object-oriented programming, it's basically that. The idea is that the cell is the fundamental unit. And every cell has an independent state or its own attributes or member data if you're a computer science person, and has its own methods or processes that can act upon its own attributes and or the attributes of its neighbors. So you have independent state, you have independent processes like cycling, death, migration processes, secretion processes. And then on top of that, we start thinking of them as signal processors, and our rules of saying this signal changes that behavior become our biological hypotheses that become encoded as rules in our agents. And so there's this kind of this one-for-one correspondence between our thinking of the biological hypotheses in our systems of what drives cell behavior directly map onto the individual cell agent rules. And so that's why when I looked under the microscope of the breast pathologist, I started steering towards agent-based models because we see this cell next to this cell, we know that there's a change in behavior, and that kind of hypothesis driven by an observation becomes a rule in an agent. So we can kind of really start matching them directly to the things we measure in the microscope. Sometimes you can measure the behaviors very directly. You can do migration assays, measure the cell linear speed. Some things are maybe harder to directly measure. You might have to infer them from, say, a bunch of measurements put together and try to minimize some kind of a mismatch. But fundamentally, it's all about one or more observations driving hypotheses that become rules, and then trying to say, how fast does a process happen? And so that's what we're doing here. And our agents really then become signal processors, that you have a library or a dictionary of signals that you're allowed to sense. You have a library or a dictionary of behaviors you're allowed to modulate, and you have mathematical formulations tying the parameters of the biological processes to the signals that they are sensing, and those become our rules. And we'll get back to that a little bit soon. And so the agents are the cells and also the software objects that can do their own thing. And so we're using this approach to build a virtual laboratory. That the first thing we knew is that in biological systems, unlike a lot of things we studied in applied math before, it's not just two PDEs, but a whole bunch of PDEs. We have oxygen, glucose, signaling factors, metabolic factors, drugs, waste products. You're very easily, in a simple model, talking about 5 to 10 diffusion reaction equations or more. And so the first thing we turn to is saying, can we efficiently solve a system of 5, 10, or 15, or 20 diffusion equations on a desktop on 2D and 3D? And so we spent a while just building a nice modular finite volume code to be able to solve that reasonably efficiently, starting on desktop processors when you are kind of toying around and building your hypotheses and say, now I'm ready to go ship it off to your cluster and do something big. But to be able to seamlessly move between, it's gone through all the typical stuff you're going to do when you do numerical analysis. We proved that as, say, a convergent and second order accurate space, first order time, if that means anything to you as a numerics person. If it doesn't mean anything to you, all it means is that you want to make sure that your numerical solution matches the equations you're claiming to solve. Because if you can't even match that, then you have no idea if your equations actually model real life, if you haven't even solved your equations correctly. So we made sure that we're solving the equations correctly, so we can say, now that we know that that's right, we can really focus on saying, are equations even appropriate to studying this biological system? And we make sure that it scales, so if you add more and more diffusion equations, the performance doesn't collapse, that you can keep adding more equations and with a marginal cost on your system. And this is open source, which means that anyone can download it and use it with a nice, easy, commercialization friendly and also open source friendly license. And it works across platforms. So, one person can work on Windows, the next person on Mac OS, next person on Linux, use the same code. And when they're ready, send it off to a cluster to run the thing. So you don't have to modify your approach based upon where you're sitting and where you feel comfortable working. So that was published a few years ago. And a key part of that was that it had the diffusion of chemical substrates like glucose, oxygen, and it had cells as point sources and sinks. So here's a cell over here. It can secrete something that becomes a source term. Here's another cell that's consuming something that becomes a sink term in that PDE. And we could basically says that if you have cells in static locations, we could solve that. So that's great for a snapshot, but not great for dynamics just yet. So we built PhysiCell as basically on top of that, saying now that we have the diffusion part out of the way, now take those static sources and sinks and move them around. and add rules to let them divide and change state. And that became the basis of our agent-based modeling platform. And we started building built-in processes, built-in diffusion, cycling models, built-in death processes. So we have built-in models for apoptosis and necrosis, just already built-in models of division, so you don't have to like rewrite this every time you build a new model. And that was something that actually differentiated us from a lot of other agent-based modelers who said, oh, well, why would you want to do that? Every modeler wants to write their own model. And so everyone ends up writing the same model over and over and over again, debugging it over and over and over again, slightly incompatible. You can't be sure if they actually are talking about the same thing. And that's a problem. So you spend all your time writing low-level processes instead of getting biology done. And so we said standardized cycle models, standardized death models, standardized cell Brownian motion models, and things like this, adhesion models. So you can say, if you tell me what a process is, it will mean the same thing to anyone using this platform. And you focus not on coding those processes, but saying, how often do you do those processes and how fast? So we can focus more on the biology side of it rather than the fundamental operations side. So this is also cross-platform open source. So it runs on any number of platforms. So we typically will have teams of undergraduates writing code in C++ in this, and Windows on one laptop, Macintosh on another laptop with shared GitHub repository. And then when we're ready, we ship the code off somewhere and explore it and run 1,000 or 100,000 simulations on a cluster somewhere. So that works out very nicely, that you don't have to worry about that. And this also scales very nicely because of our cell communication has finite distances for the contact interactions. It means that the code and the cells all have finite non-zero size. It means you can put bounds on the interactions and it scales linearly in the number of cells. So that's actually really important that the cost doesn't blow up as the number of cells goes up. So we put these together now, and now we have the diffusion parts of our laboratory, the extracellular chemical environment, coupled with the cells and their individual processes that gives you the biology part of the environment. And together that forms the basis of our virtual laboratory. And we can spend all of our time saying, let's come up with our hypotheses and build a model, watch it, compare it to real life, and then keep iterating to see if we can find the right hypotheses. And so that really is a thing, that our typical process is we sit down with our biologists or clinicians or other collaborators, say, what's important in our problem? What are the important chemical signals that you think are happening here? What are the important cell types that happen here? We have some notion of the cell rules, the biological hypotheses that become our agent rules, and we'll start coding these up and building models that allow us to ask these what-if questions, like, first of all, if these hypotheses are reasonable, what are the emergent behaviors and are they reasonable? And once you get to that point, say, what happens now if I knock down some interaction or increase some interaction, introduce a drug that ramps up proliferation or ramps up cell death or changes migration or changes secretion of a chemical factor? If I can affect any of these behaviors, we can look for the emergent systems level effect to see if it gets towards our desired community effect. And so can we then steer that system? So I want to give kind of a handful of vignettes and then transition to some of our newer work that is, kind of goes back to, I think, some of the shortcomings of traditional agent-based modeling. And I think computer modeling, mathematical modeling in general can be very difficult for multidisciplinary teams. We're trying to find ways to streamline that process now. So our first step is actually looking at what we call phenotypic persistence in breast cancer. Danielle Gilks, a collaborator of ours at Johns Hopkins, has this beautiful marker system that she developed where the cells are engineered to originally, these breast cancer cells will fluoresce red. And then the presence of hypoxic gradients, and they get into low oxygen zones, the HIF1 alpha will trigger these cells to basically snip out the red gene and then allow a green gene to permanently enable. So this allows you to track the fate of any cell that is either currently or has at some point in history been hypoxic. And so this allows you to ask a wonderful new set of questions. For example, if you have a, one question I actually had at one of these PSON meetings a long time ago is I took an informal survey of the biologists across this meeting and said, imagine that you have a hypoxic cancer cell and it's adapted to hypoxia, but doing various things like increased motility, glycolysis, different adhesion, different proliferation. Take that hypoxia-adapted cell and plop it back into normal oxygen conditions. Will it return to its old behavior before the hypoxic adaptation? And if so, how long will it take to adapt, to revert back to its old thing? So what's its phenotypic persistence of its new adaptation? And basically, I got answers ranging from it will never adapt to it will adapt instantaneously and everything in between. So the answer from my survey was the whole right half of the real line. So that's a fairly open question. And we had no way to test it until we ran to Danielle with her amazing fate tracking system. We could finally have a system where we can test these kinds of questions. So a question with an appropriate technology, with a mathematical model, lets us start getting into this kind of fundamental biology. And so our model, we try to model this fate mapping system. That we have our virtual cells have a virtual red gene, and a little tiny ODE model says in hypoxic conditions, you have a probability of switching genes to turn on the green and turn off the red. And in addition, she had experiments where we could say measure proliferation of the red cells, measure proliferation of the green cells, measure migration of the red and the green cells, and use approximate Bayesian modeling to computation to infer some of the stochastic properties of the migration. And so we could model this and then have one sliding parameter that says, how long does it take to revert behavior once you escape hypoxic conditions? So we could take that number line properly and actually put it into our model. So take that and start with, an initially normoxic spheroid, grow it under the oxygen gradient so you get decreasing oxygen inside the center. And eventually, and we could vary that parameter, the phenotypic persistence time. So the first thing we did was a zero phenotypic persistence, which means that the moment they get out of hypoxic conditions, they get out of trouble and they revert back to their old behavior. And as you might expect, they form these concentric ring structures that say any cell that's trying to escape trouble zone will migrate just enough to get to the very edge of the trouble. And then they go back to their old behavior. So they have no reason to continue migrating. And then, of course, the tumor grows a little bit bigger, which means the trouble zone has migrated outward because the oxygen gradient changes. So they migrate a little bit further, and then they stop. And so these concentric circles grow for all time, and no green cell ever breaks out because it has no reason to. If cancer did that, it would be fantastic. You guys would be out of a job because there would be no invasion, no metastasis, job done, we can go home. Unfortunately, that doesn't match, right? So we know that for at least a lot of cells, I seem to have caught a mouse over here. Okay, so if you have an escaped mouse from your model system, it's over here. So that unfortunately is not the truth. Now if we slide our slider bar and say if the persistence time is finite or infinite, now you get interesting emergent behaviors. So if you zoom in here, and it might be hard to see on the screen, so we'll go to another screen in a moment, you get these plumes of green cells emerging into the red. This is kind of an interesting thing because prior to Danielle's imaging system, those structures were invisible, except maybe in Bob Gattenby's GLUT1 marking stuff. But those were previously invisible structures. And now we have a model system that can actually create them de novo and say, we have a set of hypotheses that can create them. The interesting thing is you see these green plumes forming together, and the first inclination say, oh, that cells moving together, there must be collective migration. There must be some kind of coordination here. But it turns out that simpler models can explain it. You don't need collective behavior. One cell stochastically finds a weakness in the material, migrates through, it widens it out just a little bit, and the other cells take the path of least resistance and get this kind of reinforcing pattern of a green plume, and they invade out until they reach their persistence time, and then kind of stop and start proliferating again. And that's what happens. Now, if the persistence time is permanent, instead of just forming a plume, they'll go out and continue migrating kind of for all time. And so you can imagine that the persistence time kind of says how far out that plume can grow before they stop and do things. So that's what the model predicted, just sliding that bar. And then experimentally, I'm sorry.
Is green metastatic?
So green is basically can be metastatic, right? It has that early invasive potential. And if you had more hypotheses, it could go and form a metastasis.
I want to make sure I had defined that.
No, it's a great question. Yeah. So the green ones are the invasive ones that have the greatest likelihood of metastasizing. It turns out that both red and the green can metastasize, but the green ones go first. The interesting thing is in a mouse system, we found the same green plumes. And the way I like to think of this too is, we built our basic model with basic hypotheses, explored it, and said, if these hypotheses are right, you form this structure, and then independent observations that were not used to build our model. We actually had no idea about these plumes when we built our model. We said, oh, our model's acting weird. I hope this isn't terrible. And said, oh my God, no, we see this in our mouse. So that's like good news. But that's the idea, right? Is that you want to have a basic model that you can explore that generates testable hypotheses. They validate against independent observations that weren't used to build your model originally. So this is kind of like having training data and validation data. Well, here you want like training hypotheses and validation hypotheses in a sense, right? Or training observations, validation hypotheses. Don't use all your observations to build your model. See, hold some back that you haven't seen yet and see if your emergent behavior matches those. And that's kind of like the logical equivalent of training and validation. And so we built this up, and it turns out the same behaviors happen both in 2D and 3D, which is always nice. And we do have a cloud-hosted version of this model that you can play with if you want to try it out on your own. So there's one example of kind of a vignette of taking this virtual laboratory system and exploring it to generate real hypotheses that then get validated by the actual biological model system. And so, and I think that's nice, again, that you have a novel experimental system that makes a structure visible for the first time, and a mathematical model that goes with it that can explain the new structures you've just seen for the first time. And to put them together, I think that's really a powerful combination. Now, the next model, so I was asked to talk a little bit about metabolism, so I thought I'd pop that in. So these are really fresher slides, but we have this great approach in our Yule one, the CSBC, with Shannon Momenthaler and Nick Graham and Stacy Finley at USC. And the key question here is that cancer cells and cancer-associated fibroblasts, these are kind of these stromal support cells, are known to exchange metabolic products. And it's thought that exchange of metabolic processes, products, where cancer cells are feeding fibroblasts, fibroblasts are feeding cancer cells, this kind of cross-feeding idea somehow enables the cancer cells to become a little bit more malignant and a little bit more survivable and a little bit more resistant to different cancer therapies. And so we're trying to understand how the exchange of metabolic products in this community might be driving resistance, or at least to drive the growth behavior. So the approach is that Shannon Momenthaler is growing these cancer organoids and also 2D cultures in a high throughput system with controlled oxygen conditions, particularly in colorectal cancer for this particular project. So she can run these high throughput wells, take all sorts of automated images and get data, and also sample the products and then send them over to Nick Graham, who's running the mass spec to measure all the metabolites. And then Stacy Finley is a modeler over at USC who does FBA, so metabolic modeling, flux balance and balance analysis models. And so she takes these detailed measurements of metabolites and also the community growth rates and builds these single cell flux models for the individual cell metabolism. And then our job is multi-scale integration. Say, now we're going to take these cells, put a copy of that metabolism model in each of the cells, model the movement of metabolites in the 3D environment, and see if it can predict the emergent 3D behavior. And it turns out those, here we are in year 5, and we're getting it to work. So, so first, creating the metabolism model, this is not my specialty, so I want to apologize a bit for that. But the basic idea is that it's solving a massive constraint optimization problem that is a bit underdetermined. You know some structure of the metabolic reactions, and you put that into kind of a logical structure. So FBA models take all the time dynamics out. The good news is there's no time dynamics. The structure of the model is enough to start understanding and balancing things, and it's easier to calibrate and match to data. The bad news is you lose all the time dynamics in FBA models. So it's a pros and cons thing. The idea is that you measure the population of cells at the beginning, at the end. You assume, and this may or may not be the right assumption for cancer cells in particular, that the goal is to maximize biomass creation. And so subject to the constraints of available metabolites and of fitting the data and of the known interactions in these stoichiometric matrix, you come up with the best possible fit to the information you have to come up with a flux balance analysis model. And to run this model, you're solving constraint optimization problems. So you're trying to fit your inputs, your outputs, and match the balance of fluxes as they're known to propagate through that network of interactions. And so Stacy let this work and published a beautiful paper that gave us kind of a starting metabolic model. So that's available, looks like in Metabolic Engineering, published a year or so ago. Now we want to do the multi-scale integration. So now we have, let's talk a little bit more about the experimental system. It's actually a big 3D, at least as far as we're concerned, it's a huge 3D system. You've got a lower region that's filled with matrigel and cancer organoids. It could be dozens or hundreds of cancer organoids seeded into that matrigel with a big reservoir of fluid on the top. So you can imagine oxygen can diffuse from the top in. Everything else is whatever was in there at the beginning is being consumed and moved around. And so we have a lot of diffusion equations to solve in a really big domain. And so that's one challenge, is just the sheer scale of that well in hundreds of micrometers, cubic microns, sorry. No, actually cubic millimeters, even worse. Solving that humongous system of reaction diffusion equations efficiently with all the cells inside it. And then the next thing is we want to integrate this FBA model in each one of our cell agents to say that they all have their own copy, their own individual metabolism, so they can secrete metabolic factors. So diffusion gets it to the cells. We put that into the FBA model. That is supposed to then predict the metabolism and the secretions and the growth rates of the individual cells. We kick that back up in the environment and then do the feedback that would be emergent through this diffusive interaction of the metabolic products. Well, FBA is a constraint optimization problem. If you have 100,000 cells and my time step is 0.01 minutes. That means I have 100,000 constrained optimization problems to solve every 0.01 minutes between now and say 14 days. That's computationally a little difficult, as it turns out. There are things that we had to come up with solving that challenge. And then just the mathematical challenges of interfacing these two models. The FBA is very much a static model. And so it kind of turned into a quasi-static model saying, given this current state, solve the optimization problem, and then we give the growth rate. So we kind of added time development back to it, but just interfacing the math turned out to be hard. So first, let's talk about the efficient solution of these FBA models. Well, the nice thing about them is that you're basically solving all of them to steady state. And we're going to have some kind of big input space and available substrates and the cell sizes and things like this. And so what we could do is we could take that model and kind of run it offline. So we ran hundreds of thousands of copies of the FBA model to steady state to say, as a function of my inputs, what are my outputs for my biomass creation rate and secretion rates? So 100,000 model runs in MATLAB doesn't take that long to run, but you wouldn't want to run that all day long in your simulations. So we just use that to generate training data. And then we took a really simple deep neural network structure, say take 80% of the data, use that to build our deep neural network model, take the other 20% and make sure that the R-squared score is basically a gigantic nonlinear regression, make sure that it fits well. So, okay, I have a really good DNN approximation, it's called a surrogate model, to stand into the place of the full FBA model. So now instead of having to run the FBA model every time, I can say I pre-ran it a whole bunch, and I trained my surrogate model to take its place. And the nice thing about deep neural networks is that they take a long time to train, but they don't take very long to evaluate. So they're a really good acceleration. In fact, in our testing, compared to the original MATLAB code we got from Stacy's lab, running the same number of evaluations of the deep neural network in C++ in our agents compared to the original MATLAB model is about 15,000 times faster, which means it is way more feasible to use this surrogate model in this place. And so we found that this now is zero problem. You can put in a copy of my approximation of this metabolic model, 100,000 cell agents run in 3D. This is not a limiting factor in the simulations. This is basically a little tiny bit of extra computation time on top of all the other more difficult stuff like diffusion. So now it's actually practical to put in a complex metabolic network and approximate it and put in each individual cell agent with their own states. And because we didn't really, yeah, question.
When you put the training set for your DNN here, is that the original FBA model ran with exploration of all parameter space?
We didn't explore all parameter space, but we did explore all input space. So we know that across our domain, they're going to be in different metabolic conditions. That they'll be, say, this one will have this much glucose, this much glutamine, this much lactate, this one will have a different amount. So we varied that input space.
There are no parameters in FBA.
Well, so we assumed that Stacy already built the FBA model and got everything else parameterized, and that we only had to vary the inputs. But even varying the inputs is still a constrained optimization problem in FBA. But at least we didn't have to train everything we built.
Are you just working with the external fluxes?
Primarily working with the external
Yeah,
fluxes.
you run the FDA and train your model with the external flux.
Yeah, that's right. That's right. Saying that the internal flux is already balanced by the previous step. And then for that pre-trained model, we solve the smaller, but still, constrained optimization problem for the external fluxes. Exactly.
How frequently are you tuning that approximation in terms of the variability of each of those parameters? So
That's a good question. So we took a look at the boundary conditions, what we expect to see at the highs and the lows, and use that to kind of constrain our search space for the inputs of glucose and all the other fluxes. And we did, part of it we haven't shown here, is we did a model matching to say, the inputs in terms of dimensional analysis, the expected inputs for the FBA model have certain rates in units, right? And that we could approximate that by matching it with the source sync terms in our PDE model to kind of come up with this model equivalence of the output of our model becoming the input of theirs. And that helped us figure out the scales of the input values to try in the training. And then we really super dense, I think we did like Latin hypercoup sampling, basically to sample that input space and generate the training data. But, so it turns out, it was actually very disappointing. It turned out to be super linear. We probably could have just done linear regression. I was actually really disappointed, but the technique we built could approximate any number of complex intracellular models with the same approach. As long as you expect it to go to a quasi-steady state compared to the timescale of simulation, it should work well. And another nice thing is, in the past, when we had to build a plugin to an intracellular model, We had to look at their code, figure out how to run it, how to compile it, how to link it to our code at runtime and do all the debugging. This means that because I can just approximate that model and never have to build that interface again. I just say, if I can approximate your model, now I have a built-in interface for any deep neural network, and it should work great. So multi-scale integration to new plugins should actually be a lot easier with this approach. So let's
Do you see this type of stuff being popular? Using like
It should be. Well, we're working on a preprint on this now. I got to say, the biggest challenges for us was figuring out the math of matching these FBA models to the dynamical inputs of our model. And then to say that on the, so that's actually is, I'll skip this slide and come back to it. But then we have to figure out the interface mathematics.
But that's
Right.
the power of what you're doing is you can also modularize an FBA model and use the DNN for the parts that you don't want to study.
That's right. That's right. Anything that's that we can't approximate away or pre-learn or fit, yeah, we can definitely do that and get away. Oh, so I'm looking forward to our meeting. I remember seeing you on the agenda. Cool. Yeah, absolutely. So we can abstract away things, and we don't have to write custom code interfaces for every new thing we want to do at the intracellular scale, which is appealing because that is so much software engineering. I mean, that basically took a whole bunch of software engineering to figure out. Well, first we were trying to directly do the constraint optimization until we realized how stupidly expensive that was computationally. And then said, well, that's it, we're frustrated, approximate the DNN, oh, thank God it works. And so that's kind of where we ended up with it. But no, we were actually, I mean, a priori, we didn't know it would look so linear. We probably should have known if we'd done more analysis. But the approach should be very generalizable to other complex intracellular models. So it could also be very modularized that way. And so then the interface says that we had, in our reaction diffusion equations, we have sources and sinks for the cells, and those need to match the expected dimensions and time spans of the intracellular models. Like, okay, I'm going to run the intracellular model for 0.01 minutes. I have this amount of substrate externally available at that cell's location times this delta T times the source rate becomes this amount of stuff that's fluxing into the system on the one end. So I can kind of do this model matching to get there to map extracellular conditions at the cell's location to the expected inputs to the FBA model. And then the other side was saying that the FBA model would predict a certain biomass creation rate, which we had to turn into a cell volume creation rate by by a model comparison. And then the cool thing was that we would override the built-in volume model of our agents and get it directly from the FBA model. And whenever the cell would double in size, we just divide it then. So the interesting thing about this is that this is auto-calibrating the cell division rate without having to directly calibrate it to something like K67 or other cell cycle markers. And the other thing is that we're building the FBA model to high throughput 2D experiments and then saying, well, those reactions ought to hold in 3D. And so we take the same model and put them in the cells into 3D geometry and see what happens. But it should auto-calibrate the proliferation rate even in 3D without taking additional measurements. So that's kind of the power of those, to take cheap 2D experiments to build your metabolism model. And then this upscaling technique gives you the proliferation rate in other geometries other than just 2D. So that's the idea here. So the last thing is we have to do the 3D diffusion efficiently. Usually we were just using uniform 20 micron mesh because of the diffusion length scale of oxygen and glucose and these things. But if you have a 364 cubic millimeter well, that's like 6 billion voxels. And our system likes to have 1 to 10 million voxels, which is a bit of a difference in orders of magnitude. Fortunately, most of the action is happening in the matrigel. That's where there's the cells and the substrates and the gradients. And then once you get into the fluid, it's mostly just a vertical gradient. And so the way we did it is we basically divided this into 2 domains. We did the matrigel region. So I should point this way for our studio audience, not remote studio audience. Here's where we need diffusion in 3D. Here's where we only need diffusion in 2D. So here we have cheese cubes. Here we have bread slices for different compute domains. And then we just did a very careful treatment of the fluxes across the shared boundary. So you could solve this, a little bit of diffusion in 1D here. Sorry, a little bit of diffusion in 1D here. A little bit of diffusion in 3D here, do some manual fluxes across the boundary. And if your time slices are small enough, you can get away with this domain decomposition and it still converges accurately as you expect.
Years ago, we did a finite element calculation where we solved part of that problem by defining infinity element. That basically mapped from wherever we were to infinity. And as long as you didn't ask questions about what was inside the infinity element, you get the fluxes right.
Yeah, that works well. That's basically this very similar approach. Yeah, that's exactly right. And in my old continuum days, we used to do this too, where we'd have like a circular domain where we do the diffusion and everything outside had a far field condition, and that would work really well. So this is basically allowing us to do a little bit better than just a far field condition, but we say that out here, the gradients aren't so important. We just need to match the fluxes in that reservoir of metabolites outside of the matrix where all the action is happening. And so this gets a humongous speed up and makes it feasible to simulate this entire 364 cubic millimeter well down to the resolutions we care about on a desktop workstation again. So now we can do the humongous domain. We can do all the agents. We could do that before, and we could put the metabolic model in all the agents. So the whole thing now becomes computationally feasible. And so now let's see if it works. So we did a little bit more calibration in 2D just to make sure. And it turns out that if you don't have a really basic mechano feedback, that you can still overpredict growth even in 2D. And that's part of the problems of flux balancing, is that they're not really considering the neighborhood effects and the community effects when they're coming up with their constraints. That pressure and density of cells probably are another constraint that ought to be in an FBA model when you build it. But we were able to basically look at different places where we could apply mechanical feedback. And pretty much you have this chain, right? Substrate absorption becomes supply terms to an FBA model, which becomes energy creation, which becomes biomass creation, which becomes volume creation, which becomes cell growth. So if you want to do mechanical feedback, you have to interrupt that somewhere along that chain. Most models interrupt it at the end, saying, well, if I don't have space, just don't divide. Well, the problem with that, right? Or if I have too much mechanical pressure, would be the other way to do it. Problem with that is you haven't interrupted the beginning part of your chain. So they're still absorbing substrate, they're still creating energy, still creating biomass, and still growing in volume. And now you have a bunch of cells overlapping, and it's stupidly non-physical. So that doesn't work. Now, you could intervene anywhere in the middle of this chain, but we didn't have any real evidence to point to that. So we went to the beginning of the chain and said, well, if there's too high of density, we'll just slow down the influxes of the biomasses, which means the FBA model doesn't see the stuff coming in, so it doesn't make energy, so it doesn't make biomass, it doesn't grow, and it stops growth and division. And that actually worked out pretty well. And there is some evidence that the cells do have a regulation based upon overcrowding and metabolism. Because you can imagine, you could have intermediate things, right? You could keep absorbing stuff, keep making energy, but I can't make biomass because I don't have room for it. So you just have to flare. It's like the flares on an oil well, right? You could burn off that extra energy or do something. But you need something to account for that conservation in the middle. I didn't want to introduce some mechanism that I had even less evidence for, so we just cut off at the beginning of the chain. And our growth curves in 2D were able to match very nicely with almost no tweaking of this mechanofeedback to the observed 2D growth curves. Now, that's not validation yet. That's just saying that we're able to reproduce what we saw in 2D really well. So now we said, take the same cells and seed a bunch of cancer organoids in 3D, and the matrigel and everything matches the physical dimensions now of that humongous 364 cubic millimeter well, and no changes to the model parameters whatsoever, except one thing, that in 2D, we have 6 neighbors, so we have a non-dimensional pressure that's tuned to 2D. In 3D, you have 12 neighbors in a close packing, and so we just had to change one thing, the mechanical feedback, to make it equivalent 3D model. No other changes to the model whatsoever. So we simulated a few days of growth, and our model predicted a growth rate of 0.024 hours inverse in this 3D well. The experiment said it was 0.027, so we're off by about 11%. Considering that we didn't do any calibration to 3D whatsoever, it's just emergent, we're pretty happy with that. So God help us, we will be publishing that. Next steps, we're going to add now the calves, and they will have their own metabolic models now that we know how to do all that. And then we'll have the cross change of products. The metabolites will be made by, secreted by calves and consumed by tumor cells and vice versa. And that crosstalk auto affects the interaction of the system. And then we can do virtual knockdowns of that metabolic network. So this is kind of the next step. So there's a fairly long vignette, sorry, but it says how we're taking kind of 2D high throughput measurements to build an FBA model that then we use to calibrate a 3D model. And so that's kind of interesting. Now, not everyone here is a cancer person. Actually, we're mostly cancer people, but during the pandemic, we did branch out and try some other things. And we built a large coalition of virologists, immunologists, modelers, and others to build a COVID-19 model. And that iterated over several versions over the last few years, starting with a really basic model of viral replication and then diffusion and reinfection, then starting to add on actual receptor dynamics and trafficking as the entry point of the virus, then starting to add in basic immune interactions, and then we even added in a far field lymph node compartment of ODEs to say that cells leave your local spatially resolved model, go off to the lymph node, stimulate production of immune cells and antibody, which comes back into your local model to fight the infection. So you have basically a notion of innate and adaptive immunity in this model system. So here's a typical model, or version 5 model. The blue is epithelial cells. We have a random scattering of initial viral infection that starts infecting the cells, replicating, goes out, kills the cells, and keeps propagating out. Macrophages and other things are coming in and scooping up the necrotic debris and triggering an adaptive response. Viral count goes up until finally the lymph node activates enough, brings things in, the cavalry arrives, fights off the infection. Well, you're left with a lot of collateral damage here, but the infection does get halted. So here's your initial, this is like you unvaccinated facing COVID, and you have a whole lot of lung scarring here and lung damage. So we got that dynamic. Now, the interesting thing is take that exact same model, but now you've trained your immune system, so you have a higher antibody count and your lymph node compartment has a different state than it did before, and do a subsequent infection on a new set of tissues. So you went out, you said, oh, this is great, I'm immune now, and you went clubbing and you got another infection, and you can see you're still getting some tissue damage here, but because we already had a bunch of cells ready to respond, the damage is a lot more limited this time. Now, of course, your virus mutates and this goes away, but we were able to get that this basic model was to have this memory effect as an emerging feature. So we were able to capture a lot of benefits from this. And the cool thing is, because macrophages are macrophages are macrophages, neutrophils are neutrophils to an extent, primarily differing with the frequency of the rules, not necessarily what they do, we could take the same model system and go back to cancer immunology. Say now I have, rather than responding to an infected cell, I'm responding to a malignant cell. And we can have the same ODE model of the lymph node of building up a response, and we could actually do models of macrometastases being responded to by a local adaptive immune response. And actually, depending on the parameters, yeah,
Just to ask,
please.
For your COVID model, how is that specific to a COVID infection?
That's a great question. It basically isn't. Other than that, it is an RNA virus rather than a DNA virus, which changes a little bit the structure of the basically the structuring of the viral replication model in particular. I mean, you could cross out the word ACE2 receptor and call it some other receptor, and you would basically be there again. So it is fairly generic right now. I agree. What would make it more COVID-19 specific would be matching the timescales and dynamics that we observe for COVID data. But it was initially motivated by that, but you're right, is more. And the fact that we can transfer it over to a cancer model pretty quickly shows that it's not super specific, in part because we're not modeling the molecular dynamics of the receptors and the viral binding and everything else. I'm sorry?
Didn't you have the ACE2 receptor?
Yeah, we did some really basic receptor trafficking models. We would have receptors that bind with virus that goes in, and we didn't do, I don't think we did any coarse dimerization here, but we did the endocytosis process, and then we had to have the virus get released from that receptor, and then some receptor would be degraded and others would be recycled back up to the surface. So in principle, if you ran out of receptor, you could run out of viral entry. It turns out, though, that with reasonable, like same parameter values, you would never run out of receptors in this particular case. So it wasn't really an important feature for this particular virus infection model. But in principle, you could model cases where you do run out of receptors, or receptor competition would be a really interesting thing to think about. So drugs that target receptors might have been another valuable thing to think about, for example. So the thing that was interesting, though, is to build this model, we had to basically come up with hypotheses, become artisanal handcrafted C++ applied to the different agents. And as the model complexity grew, that became really, really hard to maintain. And it came to a point where basically only one guy knew the code well enough to do any modeling. So really hard to get coalition and community participation in that process, and really hard to understand, if I add this rule here with this handwritten code, have I broken my other rules? Are they compatible? Did I forget something? Did I actually fully annotate my model and what's going on in it? So this is like the biggest model we've ever built, and it really exposed some of the limits of mathematical modeling approaches that we've been trying. And it doesn't leverage prior modeling. You have to keep rewriting this stuff. Increases likelihood of error. It makes it unmaintainable. New people can't contribute, and you have a great probability of not even fully annotating what you think you've done. So that's a real problem. So you read a lot of agent-based models, especially some breast cancer tumor immune models that we've tried to replicate. We actually can't even find a list of their hypotheses. We don't even know what they're modeling. Even in their method paper that says, we just did everything in this paper. And then you go look at that paper and say, we did everything in this paper. And then we look at that paper and read the chain all the way back, even read the direct source code. I cannot figure out what's happening in any of those models, but they look great.
Yeah.
So we want to see if we can kind of fix that. And so we're trying to make a modeling grammar that is more plain English. Say, this signal changes this behavior in this cell type. And that directly maps onto mathematics, which directly maps onto code. And that generates a model. So now your sentences are your model, not the mathematics and not the code. The sentences are your model. And we want to write in a way that adding new sentences doesn't break old sentences. So if we can do that grammar, we can change the way we do our modeling. I'd like to posit that we've done. So one thing we found is we kind of abstracted out and say, we know, for a given set of cell types and a given set of chemical factors, we can figure out what our entire dictionary of signals is. And we can write them down in a standardized language that this signal name, like oxygen, means extracellular concentration of factor X, and that maps onto something that I know how to map to into code. So if I write this symbol in a sentence, I can directly correlate it to something that the cell can access in a programmatic, easy way. So I have like a controlled vocabulary of signals. You can also auto-generate a dictionary saying that cycle entry means a certain parameter from my cell agent. And so I can read it or I can write it very easily now with a symbol. And so I have a dictionary of signals, a dictionary of behaviors, and I can put them together in a grammar saying some signal from my signal dictionary increases or decreases some behavior in my behavior dictionary with some optional parameter values, and that completely codifies that biological hypothesis. And then I can map it onto the mathematics as like a Hill response function. I have my base value of my parameter in the absence of a signal. I have my maximally changed value when you have a lot of signal, and I have a response function that says how that I map that variation in signals. This is really some of the work you guys have done on your like Hill parameters, and when you have the synergy and the counteraction models that you guys have done. So we just use these Hill maximum value effective. They're the base value and the maximally effective value. And so a simple rule would be that oxygen increases cycle entry from base value towards max value, Hill function of some half max and Hill power. And that becomes a really easy equation because I can write the equation systematically. I can turn it on to code. The cool thing is that I can turn this into both up and down rules. The one rule increases behavior, another rule decreases behavior, and I can put those together compatibly into basically a bilinear interpolation of the up and the down signals with two response functions. And it works, and that means that adding another rule didn't break my previous rule, and I can look at competing effects. That assumes that they make some certain simplifying assumptions that may not always work, but it's a start. And so here's a simple, combo rule. Sorry, I know I'm running really close to over, but we'll get to the punchline really soon. Well, I think I'm the punchline, the joke, but so here we have, sorry guys. This time the Zoom people have the advantage. It might be because I'm in presentation mode. Maybe if I get out of this.
Try to get out.
There we go. Let me go back in. Okay. But this means that you can have, basically a two-dimensional response function, right? I have pressure decreases cycling because of the overcrowding. Oxygen increases cycling because of increased energy availability, and I can get this kind of two-dimensional response function. And the cool thing is, if I only vary one factor, I get back to the traditional one-dimensional rules. It's like along the two axes here. And we went a little bit farther with it. We made multivariate Hill response functions. We can add a whole bunch of signals that make up responses. Any one of them alone can increase my response, and combos of them just make it increase faster. It's not a linear combination though, right? Because they're each nonlinear Hill functions. So you go back down to the base case, and if you only have one signal, it works the good old traditional way. We can add more up rules without killing your old up rules. So new rules are compatible with old rules. So I can integrate new knowledge and old knowledge easily. Same with down roles, and then it became the same old bilinear interpolation with the very nonlinear interpolation variables. And so I can add any combination of new and old rules, and up and down and contradictory rules to change any behavior in my set of dictionaries. So, and now that I have a fixed dictionary and a fixed grammar and a fixed mathematical mapping, that means I don't have a moving target anymore. I can start building graphical design tools to build these models. This is actually something we're going to introduce at the workshop this summer, is that we are making now a graphical editor saying, pull up a cell type, this behavior is modulated by this signal, up or down with these parameters, boom, there's a rule. It becomes a line of CSV that we parse at runtime without having to write any C++. So we think for like 90% of the rules of models that we want to build, could probably just do this now and not write any code. And because I have this fixed representation, I can auto-annotate my model. It'll spit out an HTML file in the table, say, this should go in your method section. This is my model. And it's actually annotated. So that's something we're trying to get to. So as an example, we wrote this model at Oregon Health Science University with Joe Gray, Lisa Cousins, Laura Heiser, Young Cheng, and said for a very small, well, to you guys, a small set of rules, to us, what used to be a huge set of rules, in a couple afternoons, we were able to handwrite the CSV files to make this series of rules based on their prior knowledge, their experiments, and reading the literature. So again, a variety of data sources and observation sources can compatibly come together to form a set of rules. It can integrate data-driven rules with knowledge-driven rules. And this becomes a few lines of CSV that parse at runtime, which auto-generates this model. So now we have a tumor growing, gets hypoxic, makes necrotic cells, necrotic debris, brings in the macrophages, they polarize to an M1 state, become pro-inflammatory, they secrete inflammatory signals that draw in the T cells from a naive state to now becoming effector cells, start attacking the tumors much. Now, though, eventually some of those macrophages will turn to an M2 state, which increases the rate of transitioning effector cells to an exhausted state. So it's kind of a temporary attack. But all this is just written in those handful of lines in CSV for this kind of a model. So we think we can move much faster to build models. So we think in the future, we can turn this from a months-long cycle to a minutes to hours-long cycle, and hopefully do it in real time. Say, sit down together as a multidisciplinary team, pull up the graphical editor, build your rules together, run a model, see what it looks like, and get immediate feedback and saying, oh, go run off and do your thing, debug, and come back to me in 3 months and tell me what you saw. You can say, let's run it together and say, oh, you know, that doesn't look right. Let's try this rule. Let's try this rule. We want to see that kind of that iterative process in real time so you can really get much faster in your progress as a multidisciplinary team. And I want to see the biologists building their own models, not waiting for us mathematicians to build them. That's I think, the true way to do this. We're trying to get the NIH to agree with that on maybe 24, but we'll see on that. So really, we see a lot of paths, right? That some are going to be expert-driven rules where they're going to write, say, hey, I know that IL-6 does this to macrophages or effector T cells or Tregs. And then you write a rule, and there it is. And then maybe you do ChatGPT mining PubMed, and it comes up with some rules. Well, we can slide them together because we use the same representation for both. And then we do another one, so we did some measurements in a lab, and we auto-generate a rule with some kind of data simulation technique. All those come together. New rules and old rules are compatible. So we can build up kind of like a library of rules as we go, and hopefully transfer them, that knowledge then, between other things. But because we wrote them as human-readable rules, it's interpretable. It's not just some black box we auto-fit, and then you go to a new geometry, you have to rebuild your model. So that's kind of the hope there. Let's see, I think I'm sharing the whole screen. But this is the kind of thing that they're starting to look like, where you build your simulation domain. How big is my domain? What are my diffusing substrates in my environment? What are my cell types? And I can set kind of hierarchically the different properties and set other parameters. And then right now I'm still hand editing my rules, but Randy's working on a graphical editor of this. But I write these rules in a notebook, and I can run it right here in the GUI, and then start plotting and looking at my model and analyzing, and even saying, how does it correlate with my different gradients? Say, oh, here's my pro-inflammatory factor being secreted by the M1 macrophages as they run around. And so you can look and explore these models in real time almost. This model took like 3 minutes to run. So you really can try to iterate through. So that's kind of where we're hoping to go. So thanks for letting me go a few minutes over and for exploring this together.
Fantastic stuff.
go open up for questions, trainees first. Anyone? Austin, you fall into the category.
Okay, I mean, I'm putting that sticker, so.
So, yeah, actually, we'll close it off here and then allow the students to just continue on the conversation. So